Okay. So I think we left off on Monday talking about how do we get bigger muscles, talking about hypertrophy, right? And the key thing, if you want your muscles to grow, if you want them to be bigger, is we have to make new protein. Okay, new protein, more actin, more myosin, more of all of the things that go in it. That process is called protein synthesis, or it is sometimes referred to as anabolism. Okay, and so we need to use resistance training, and at times, okay, the protein that we consume in our diet or in a supplement or anabolic steroids to activate these cellular pathways that lead to an increase in protein synthesis, that will turn on the genes in the nucleus of a muscle fiber and say, make more actin, make more myosin, make these sarcomeres and myofibrils and fibers bigger. Okay. So that's the general stuff we're going to try to get at today. I mentioned at the end, and I'm going to reiterate it here because it's going to come up. Okay. We are going to separate the protein synthesis response that happens after one bout of exercise versus the net protein kind of synthesis response that occurs over 6 weeks, 8 weeks, 12 weeks. Okay. And we've got to be very, very clear about this because you get this giant increase in protein synthesis after one bout of resistance training. And if you extrapolate that out, you would think then my muscles are going to get to be huge, and they don't. Okay. So there's a bit of a disconnect. We don't fully understand the hows and the whys of all of that. Okay. But there's a little bit of a disconnect. And I think to be an informed consumer of supplements and of how we want to train, you've got to be able to kind of have some nuance in how we look at this. Okay. So we'll start off with a very, very brief review, okay, of the idea of protein synthesis. Okay. Nobody freak out. Nobody have PTSD about like intro to bio or human phys or something. Okay. I remember being a biology major and we did transcription and translation in every class. So I've had it like 12 times. And by the end, you're like, dear God, not again. But this is all we're going to do. We're just going to talk about the general process, right? In order to make new protein, we have to tell the genes in the nucleus of that cell that code for those specific proteins, hey, we need more of this protein. Okay. So the process, okay, of taking those genes and making a molecule called RNA, which is going to be the blueprint for making that protein, turning DNA that has the genes into RNA that we're going to use to make the protein is called transcription. Okay, it's called transcription. So when we do resistance training, when we stimulate the muscle and overload it, we are trying to turn on certain pathways in a muscle cell that will tell this nucleus, make RNA from the genes that encode actin, myosin, all of the other structural proteins that will make us a new sarcoplasmic reticulum, et cetera, et cetera. Okay. So the training activates transcription. Once we have transcribed the messenger RNA, we've made the blueprint. Then, so exercise plays a role here. We then have to take that messenger RNA and it goes to a ribosome. And then we, at the ribosome, will, based upon the code or the blueprint of the messenger RNA, we will hook together amino acids to make the protein that we have coded for. Okay. Exercise works here. Diet and having protein and amino acids available works here. Okay, that's not 100% true. Exercise also does some stuff with ribosomal RNA and ribosomes and packaging and a little bit of this, but realistically, the training increases transcription. And then we need to make sure then that as we have the messenger RNA, that we have protein available so those messenger RNA can be translated into actin, myosin, and our things to make the muscles bigger. Okay. Does that make sense, guys? Do you guys understand those kind of two-tier processes? Because it doesn't do me any good, right? I eat a bunch of protein, so I've got a bunch here, but if I don't do anything to activate those genes and make the messenger RNA, I just have a bunch of protein floating around and I'm not going to do anything with it. Okay. Conversely, the reason it's important to have protein in our diet, okay, is that if we work out and we transcribe the messenger RNA, if we don't have the building blocks, then we're also then not going to make the proteins. So we have to have this balance. Okay. So when I say taking supplemental protein is probably not that beneficial for the vast majority of you, I don't want you to interpret that to mean that protein is not absolutely important for hypertrophy. It's just that the vast majority of us probably get enough in our regular everyday diet, and we don't need that supplement. It doesn't hurt anything, but we probably have enough here. Okay, but certain people in certain circumstances don't, and having that supplement will be helpful for this side. Okay. Okay. So what you saw there is protein synthesis. Okay. My dad, the former football coach, he loved a good quote, right? And I see all sorts of things now about stuff. One of his favorite things that he used to tell me, it was probably not a great thing to tell, you know, like a 13 and a 14-year-old person, is right that you're either going to get better today or you're going to get worse today. Never say the same. And that's kind of a scary thing, right, on all of this. And it was the idea of, are you going to do something in practice today to get better? Are you going to study today and get better, right? Or are you not? And that somehow implies that you're going to get worse in those ways. Not correct, right? Lots of therapy about all of that in my life. So, but it's okay. Maybe that's where some of my anxiety comes from. But protein-wise, okay, a similar thing is realistically happening with the protein and the muscles in your body right now. Protein is in a state of what we call flux, okay? And so how many of you have not had breakfast? Like me, no breakfast, okay? Many of you. We are likely, because we haven't had breakfast, in a state of what we call catabolism right now. We're in a catabolic state, which means we are likely in a caloric deficit because we haven't eaten anything since last night, okay? And one of the processes that goes on when we're in a caloric deficit is we will start to break down muscle tissue. We will start to break it down so we have protein available to then use for energy or to put into other places that we might deem more important than your skeletal muscle. Okay. So there's always some level of protein degradation in your muscles and elsewhere that's going on. So they're always getting a little bit worse. Okay. There's also generally always a little bit of protein synthesis going on. And so we have to think of your muscles and your muscle size is the long-term net balance of synthesis versus degradation. And we're just trying to balance those two things out. So when synthesis predominates over a long time, you'll get bigger muscles. When degradation dominates over time, they're going to get a little bit smaller. Okay. And it's way more complicated than that, but that's the very general big picture type. Okay. So when we get hypertrophy, positive balance, atrophy, negative balance. So then that raises the question, right? Well, how then are we regulating the signals for synthesis and the signals for degradation? Well, it's very complicated. And there's a huge number of things that go into this. Okay. Hormones play a role, although maybe not as much as we used to think that they do. Diet plays a role, especially protein consumption. Mechanical loading, so that can be walking, running, holding your body weight, or it can be resistance training, plays a huge role. Okay. Stretch as part of that mechanical loading, weirdly important. Okay. You can't do Pilates or yoga and get bigger muscles. I'm sorry. Also, if you do those, you're not going to get long, lean muscle either. That's also bullshit, right? I am never going to be long and lean of anything in my life, right? I am short and stubby and built like a second baseman with little baby T-Rex arms. My wife, on the other hand, 5'3", our waist is at the same place. She's all arms and legs, no midsection, right? She needs to buy petite pants, but they're too short generally for her and all of that. Like we're wild. So we're wondering what's Ellis gonna look like. Will we like balance Ellis out and she'll be like perfectly proportioned in those ways? It's sort of hilarious. I don't know. But sorry, I was talking about stretch and I got metabolic stress. Going to failure, metabolic stress, not running, not cycling, not that metabolic stress, but some others maybe matters. Nervous system activity. If I want to make your muscle shrink, I just cut the nerve too. Cut the nerve, muscle dies. Stop using the muscle, nerve also dies. There's this symbiotic relationship between those two. Okay. So we're going to talk about a few of these things and kind of some of the roles that we're gonna look at. Okay. So I mentioned on Monday this idea that you do one bout of resistance training and you get this giant increase. Okay, this giant increase in protein synthesis. And you may be wondering, Dr. Black, how the hell do we know that? How do you measure protein synthesis? Well, let me tell you, it's not a particularly pleasant experience. What we do is you go to a hospital, we run an arterial and a venous catheter into your femoral artery. Okay. Anybody ever seen an arterial catheter placed? I've seen a cardiologist try to do one on a girl from our lab when I was a master's student on her mom. Cardiologist did it. She practically bled out in our lab. Okay. Really, really difficult to put dangerous to put an arterial catheter in. But arterial and venous catheter, then you give people some protein that is radioactive. It's not bad radioactive, but it's got radio labels on it, so you can track it, have them eat it and consume it, do a bout of exercise, and then you just sit there and we measure and take biopsies and then measure sort of how much of that protein is fluxing in the artery versus artery, how much goes in, vein, how much comes out, and we can measure protein flux. Okay. Anybody from Texas, go to UT Galveston, go to the med school at UT Galveston. They're the single best place in the entire world that does this. Okay, and Bob Wolf is there and several other people. They pioneered this technique. A lot of what we know about it has all come from UT Galveston. Okay, so if you want to go to the beach, you can go check that out too. But here are some data from UT Galveston. FSR stands for fractional synthesis rate. Don't worry about what this is. Don't worry about what the units over here are. It's a percentage per hour of a fractional rate. You don't really care about it. It's not important what it is. Just look at the reaction of the bars. You have some fractional synthesis rate at rest. We do about a resistance exercise and we track people for 3 days. Now, mind you, they were sitting in a hospital in a bed for 3 days. So we gotta be careful about that. Because once the catheters are placed, you gotta stay. 6 hours afterwards, it's up maybe 2.5 times. 24 hours, it's up like 3 times, and then it starts to come back down. But 72 hours later, it's still up. One bout of resistance training should be sufficient to raise protein synthesis rates for 2 to 3 days. Anybody remember from personal training or personal health and fitness? How many days a week do we recommend that you resistance train? 2. So if I work out today and I wait 3 days and then I work out, logically, I'm still having an increase and then I do it again, right? And so I'm pretty close with 2 workouts a week to putting myself into almost always being here. Okay. We don't arrive at those resistance training recommendations because it was just some lark that somebody was on, right? It comes from this. Okay. It comes from the changes in what we need from a recovery standpoint and from all of this. That's where the 2 came from. That's where they do 3 to 4. Doing 7 doesn't matter. Doing 6 doesn't matter. Like, okay, you're fine, we're here, we're okay. But this is what we know from all of this. If you do the same thing and you eat a meal heavy in protein, okay? You get 2 big pieces of steak or some chicken or some brisket or whatever it is that you like, okay? Elevated for about 24 hours. If we do resistance training and then you get some protein, we can make all of this go up just a little, go up a little bit. Okay? Part of what is happening here, what you're seeing is the fractional rate. We get an increase in synthesis. You also get an increase in degradation after exercise, but the ratio of those two things gets disproportionately shifted. If you have some amount of sort of synthesis and degradation at rest, they both go up, but synthesis goes up to a greater extent after exercise. So you end up in a more positive balance. Okay. And that's what's going on with all of this. Okay. So that's protein synthesis. In the next couple of slides, we're going to talk about, okay, you're making more protein. Theoretically, then your muscle fibers should get bigger, or your muscles should get bigger. How does that happen? I want to introduce 2 concepts here, one of which we don't actually think happens, but seems very plausible. Okay, it seems very plausible. So what I'm showing you here, okay, is something that gets into an idea of a concept called hyperplasia. Hyperplasia means fiber splitting. So what that would mean is basically your muscles get bigger like bacteria or something would multiply, like mitosis in that way. You have one muscle fiber, we work out, protein synthesis goes up, I make new things, and that one fiber then becomes 2. So now I have a bigger muscle because I have 2 fibers instead of 1. Okay, very plausible in the world that could happen. We have almost no evidence that this happens in us. Okay, that it happens in us. It's really difficult to study. Anybody want to let me dissect your bicep and then put it back together and put it in and then work out, and then we'll do it again and we'll try to count the individual fibers and hopefully we get the numbers right? No, of course not, right? It's ridiculous. So what we know in humans is you take a cadaver and you find a cadaver that did resistance training, and you find a reasonably similar cadaver on a bunch of things that didn't do resistance training throughout their life. You look at their muscles and you count the fibers, you try to figure it out. And what we think from all of that is that fiber splitting probably doesn't happen in people given the ways that we exercise and train. Now, in animals, we can force this to happen. We haven't talked about all the animal models. You take a grad class with me, talk about all the animal models of how we get hypertrophy. One of the things that they do is they will go in, they cut the tendon on them, and then they stretch the muscle out. They reconnect it and stretch the muscle out to do all of this. And then they send them back on out of their way. It's called a chronic stretch model of getting hypertrophy. You do all that, you chronically stretch it, and then walking around with a very, very stretched muscle, it gets giant. Okay, it gets really big. Other things they'll do is like, oh, we're going to go in, we're going to cut your brachioradialis and a couple of things, and you just got to use your bicep. We cut the synergist muscles and things as well. Or we will cut 2 of your 4 knee extensors and just have you walk around with your other 2. Muscles get a lot bigger. When we stretch the muscle in an animal and you count the fibers, we do seem to get some splitting. So stretch is really, really weird and really important. Okay, really fascinating. We don't understand all the effects and why stretch does what it does. But chronically stretching in that model of trying to induce hypertrophy seems to induce some level of hyperplasia in animals. But we don't ever do that. Like that never happens to us. All of your Pilates and yoga and whatever stretching you want to do, I say as the least flexible person in the world, can't bend over and like touch my toes and everything. We don't do that, right? Like we're never that stretched. We don't think that this is how we get hypertrophy. Now, a little bit of evidence in people that have taken steroids that may be one of the ways that you get hypertrophy or an additional amount of hypertrophy from taking testosterone. That may induce some. So not a lot that's going on. We don't really know. So if it's not splitting, what is happening? Well, we get, in this instance, 4 myofibrils, and all 4 of them get bigger. Okay, you're not gonna go from 4 to 8, we're gonna go from 4 that look like this to 4 that look like that. Okay, and the key thing that happens here, okay, if all that is happening is we get more actin and myosin and things in a sarcomere, so a sarcomere gets bigger, so that myofibril gets bigger, so the fiber gets bigger. We're making more protein. The fibers get bigger, but we also see an increase in the number of myonuclei that are going to go into all of it. And the book goes into a little bit more depth about this, but one of the things that we think that happens when you resistance train, and one of the things that becomes so important about satellite cells and the hormones that activate satellite cells is that they help us gain myonuclei. Okay, we can only have a fiber that or a myofibril that is so big. It can only get so big based upon the ability of the myonuclei to be able to regulate and supply the things that they need. So the fibers have a limit of how big they can get before they're too big to not be. These two things string them together. So there's 2 points that I want to make here for you guys. Okay, 2 points that I think are really interesting and deserve a little bit of thought. Okay. We think we know, say in steroids, every one of you has some genetic limit on how big your muscles are going to get. Okay. Some of you, as we know, can train all you want, and your muscles might actually get smaller. I'm sorry. Okay. Some of you may get really big. You all probably have that friend who looks at the weight room and they're just swole. Okay. So what we are trying to understand as exercise physiologists, as muscle physiologists, is genetically what is it about some people that they're predisposed to being able to get bigger muscles, to respond to that. And part of it may be the number of muscle fibers that they have and kind of the starting size of their myofibrils and the number of satellite cells and myonuclei that they have. It's not even a genetic thing. So if you don't add fibers, if you're a person who has a large number, a disproportionately large number of muscle fibers in a muscle, but they start off very small, okay? And you're a person that has a sort of disproportionately high number of myonuclei per the size of your fibers, you may be someone that has the genetic potential, if you've got more fibers and they're all small, to be able to make them bigger. And you've got already have the myonuclei to support their ability to get bigger. So that's these 2 things, right? If you're a person who starts off with not that many fibers and they're already pretty big, right? Then, and you don't have that many myonuclei or the capacity to increase them, then that may set some limits. We don't have a lot of scientific evidence of this. It's almost impossible for us to test this, right, in people, but there are things that are out there that people are talking about, that maybe this is a thing that's going on. And so I just want to bring that up and just kind of give you guys a thought experiment as it goes. But we think that this is how we get hypertrophy. You don't get new fibers. The ones that you have are going to get bigger. And they can only get so big based upon the amount of myonuclei that are able to kind of regulate them and keep them healthy. Okay. Now, after cognition to contraction, you guys have probably figured out my love of a good signaling pathway. I'm not going to make you guys draw this out. Okay. No, we're so bitter that you learned all of cognition to contraction, and I didn't ask you to just write every bit of it out in the midst of that. So I thought I was being kind by not giving you like a 30-point question in doing this. But what you see here, and this was published almost 15 years ago, so you can imagine that it's somewhat out of date. We're always behind, is just a fraction of what we think the intracellular signaling pathways that lead to transcription of messenger RNA from the genes that we need to increase muscle size that are going on. You will note up here, this is a muscle cell. We have receptors on the sarcolemma, right? You've got androgen receptors, things for different hormones. You've got insulin receptors. You've got receptors or our ability to bring amino acids in. Leucine is a particularly important amino acid. What I want you guys to take away from all of this, very, very simply, is this right here in the red box. Okay. If we want to activate transcription and translation of protein in your muscles, we need to turn on a gene or a receptor here called mTOR. Okay. Everything else that is on here essentially has to get passed through this mTOR regulatory pathway right here. mTOR stands for the mammalian target of rapamycin. Mammalian target of rapamycin. This is a protein or a receptor that responds to an antibiotic. That's how we found it. Okay. And I'll show you guys a little bit there. Exercise activates mTOR through a very, very complicated series and cascades of events. Okay. You get some hormones that are going to be released. They do all of that. We activate this thing here with stretching and loading on the muscle called AKT. That activates some kinases and shit happens and we get mTOR. Okay. mTOR activates something called P70S6 kinase. Okay. Kinase puts phosphate groups on. That phosphates a bunch of things and it gets inside the nucleus and it says, hey, activate these. All I want you to know is mTOR. Although it does have some fun things like Raptor is a thing that regulates. mTOR is another one that's called Regulator, which regulates Raptor and all of this. There's some fun there. Just want you to know mTOR. Yes. Mammalian target of rapamycin, which I think is on a slide a little bit later on. Okay. Okay. So I'm going to tell you something here, and then I'm going to tell you that it probably doesn't matter that much. Okay. So I want you to appreciate the nuance of what's going to happen when we do all of this, because I think it's fun and it's cool and it's interesting and that you guys will appreciate it, but take it with a grain of salt that it's there. Okay. So people are always very interested in anabolic hormones. And we know that you need to initiate muscle growth, you need to have some anabolic hormones in your body. Okay. We'll talk about their overall general importance in a minute, but you need some. What we know is that generally that testosterone, growth hormone, and something here, IGF-1 stands for insulin-like growth factor 1. Okay. Insulin-like growth factor 1. That these hormones, if we just give them to you, in general, promote protein synthesis. Okay. We know a lot about these things from people that don't have enough. If you have what are called hypogonadal men, or men that have low testosterone levels, okay? If we give them back testosterone to kind of a normal physiological level, they will increase muscle mass, they'll increase protein synthesis. The few studies that we have where we do a supplement in eugonadal or men that have a normal amount of testosterone, they're going to get like a 20%, 25% increase in muscle mass with no training, and it's way, way better when you train. Testosterone is very, very powerfully anabolic. Okay, growth hormone. We know that if we supplement you with growth hormone, you get an increase in muscle mass, strength, protein synthesis, and it also really regulates decreases in protein breakdown. Weirdly, if you supplement growth hormone with training, you don't get any additional gains. We don't fully understand that. What growth hormone seems to be really good at is to metabolizing fat. So bodybuilders will, what they call stack, they'll take things together. They'll take testosterone, they'll take growth hormone. The growth hormone will in some ways helps them sort of lose excess body fat. One of the things is it promotes lipolysis in those ways. It's difficult to extricate what growth hormone does versus downstream. Growth hormone leads to the production of IGF-1. There's some regulatory things here. IGF-1, you can buy it. You can get on Amazon right now and theoretically buy some IGF-1. I wouldn't recommend it. Okay. Insulin is anabolic. IGF-1 binds to insulin-like receptors. It is also anabolic. But you make IGF-1 in the liver, you make it in the muscle, And then there's a special version of IGF-1 that's only made in the muscle. And it's a special variant, and we call it mechano growth factor, right? NGF, or mechano growth factor. The reason that we highlight mechano growth factor is this is the thing that activates satellite cells. Okay. And you work out a muscle, what muscle gets bigger? If I do curls, what muscle in my body is likely to get bigger? What about my quads? If I'm doing curls, are my quads going to get bigger? Probably not. Okay. One of the reasons that we get this level of specificity, you release testosterone and growth hormone and liver IGF-1, and it goes everywhere. Okay. But one of the reasons that we get growth only in the muscles that we're using is I use those muscles release mechano growth factor, and it is what's called an autocrine, and it circles back and it binds right back on that cell that just released it. And it activates the satellite cells in those particular muscles. And so this one is really, really important. People will try to supplement with IGF-1. If you want the IGF-1 supplement to work, you have to work out and then inject it directly into the muscle that just worked out. Okay. Go look at the guy whose biceps exploded. Seriously, that's why this is important. Okay. So 20 years ago, when I was learning about all of this, we thought you work out, you get this acute increase in anabolic hormones afterwards. That is part of what's driving this increase in protein synthesis. Turns out maybe not. Okay. So I was taught this, and I'm going to mention this because I think it's important, but then we're going to talk about why a little bit, why it's not maybe what we think now. So when you do resistance training, you get a bigger anabolic hormone release than doing nothing or doing like aerobic training. And we know that it's related to force production and stretch and the total muscle mass that you use. So squat, you get a bigger hormone response after squatting versus doing curls. You generally get a bigger hormone response if you do concentric eccentric movements than if you do concentric only, but nobody ever does concentric only, or if you do isometric only. Okay. So there's something about stretch and load that are really, really important. Okay. Those tend to induce mechanical stress along the membrane of the muscle, and that tells us to release the hormones. And it also tends to upregulate on the cells receptors for those hormones. So it's one of the things that happens. You work out, stress the body, you release the hormones, but the muscles that worked out then make more receptors for those hormones that theoretically make those muscles that you just worked out more sensitive to those hormones, and that's why they get bigger. Or at least that's what I was taught. You can still see that out there all over the place. Okay. Anybody a member of the National Strength and Conditioning Association? You ever want to like train to get your CSCS or anything like that? Okay. The entire chapter on how to do resistance training in their book is based upon a study that's a lie. We now know it's right. There was one study, where they did this idea that you're going to train for strength or endurance or hypertrophy is all bullshit, right? All crap. All training can increase strength. All training should, if done to failure, can increase in muscle size. But their thing where they quantify hypertrophy is do short rest periods. Rest 30 or 45 seconds in between sets. Because if you do that, you get more growth hormone release. One study showed that. More growth hormone release if you do short rest periods. And so that's how we should train for hypertrophy. Who cares that all of the evidence, when you actually train that way, it doesn't look like it's any better. But that was what all of that was sort of based upon. They've changed their tune a little bit. And this was from one of the most famous, one of the best exercise physiologists in the world. We didn't know any better. That was the data that we had at the time, and we now know differently. Okay. So here's the thing. We have some more recent studies that increases in circulating anabolic hormones is not required for resistance training induced hypertrophy. They have done some studies where we can get hypertrophy without getting this. Okay. We know that insulin-like growth factor, they don't have testosterone on here, but insulin-like growth factor testosterone and growth hormone lead to mTOR activation. Okay. Acute resistance training gets us relatively small increases in these things. Okay. And so, but because that thing is so small, we think that it's not absolutely necessary. I've done some things that block some of it and we basically go. So the going theory now is that the acute change, as long as you have insulin-like growth factor, as long as you have growth hormone and testosterone and estrogen, that acute change after one bout of exercise is not really linked to how much we're going to get. Okay. That's not to say that some of y'all have higher resting levels of testosterone than others, and that will tend to predispose you to having bigger muscles. Okay. We know all of that very, very clearly. So keep all of that sort of in there. Satellite cells are really important. Okay. We know that we need those to form new myonuclei and to be able to be there to support an increase in fiber size. Okay. That's the big issue. And so we need to be able to find a way with resistance training to activate the satellite cells and to tell them to turn on and make new myonuclei. Okay. Otherwise, we're going to put a cap on how much protein, extra protein synthesis we're going to be able to get. Okay. Well, how do we know this? Because of animal experiments. Okay. If you take in isolated muscle fibers that were growing in a petri dish, okay, and we block the ability of the satellite cells to function, you get no hypertrophy. They then took that and did that in, these are data from mice, 4 little mice. Okay. What they did was they zapped them with radiation and killed their satellite cells. Then they went in and they cut their synergist muscles and they're like, okay, we're going to cut your gastroc, walk around just on your soleus, right? And that should make that muscle get bigger. And so what you're going to see here is irradiation plus what they're going to call synergist ablation. So this is our model of resistance training, but we killed off the satellite cells and their muscles got smaller. This one is just the model of resistance training, just ablation. So you have functioning satellite cells. Okay. And we do this model of resistance training, the muscle gets way bigger. Here we just zap them and they do no training, right? And the muscle gets smaller. So what we can learn from all of this is what's called a reductionist model of human research, is satellite cells are wildly important. If you knock out the satellite cells, then even if you do good sort of well-programmed resistance training, adequate amounts of protein, we're not going to get an increase in muscle size. So they have to work in order for that to be able to go. Okay. And one of the things that we know then is that part of how we activate satellite cells is also through mTOR. mTOR drives protein synthesis. It also helps us drive satellite cell activation. More data, I know, really doesn't matter. Okay. I'm showing you guys this because the woman that published this in 2000, her name is Sue Bodine. Sue, we paid her a shit ton of money. She now works in Oklahoma City at the Oklahoma Medical Research Foundation at OMRF. And so she is one of, if not the leading experts in the entire world on the cellular regulation of muscle mass. Okay. Very, very highly cited, very widely funded. And so she's here. You guys could go see her. You guys could go work with her in the summer or go do a rotation through her lab or something else. Okay. But she was not the person that discovered mTOR, but was one of the people that discovered that mTOR is very, very important in resistance training induced muscle hypertrophy. Okay. And so they do this, they're calling it compensatory hypertrophy. Again, it's animals. They're doing the thing to try to make their muscles bigger. And they did 7 days of training, 14 days of training. The big deal is that in the gray bars here where they're not getting an increase, they take the muscles and they feed these animals the antibiotic rapamycin. And rapamycin binds to the mTOR and blocks its function. So when we block mTOR, nothing. Okay, no increase in muscle mass. And then they look at this, and all they did here was they took those muscles, they dissected them and they looked at them across the different kinds of muscle fibers and basically fast fibers, slow fibers, right? What you're getting is similar sort of blunting of an effect in both facets. That's all this is. So all I want you to know is that if we block mTOR, we block hypertrophy. Okay, that's it. How does exercise therefore stimulate mTOR? We don't know. We'll figure it out someday. And it'll be a Nobel Prize worthy thing, but we don't really know. Okay? We don't. We'll do the last slide or the last couple of slides on Friday. We'll start metabolism. You have a reading assignment for Friday. I will see you all then. If I don't have your test, I've got to have it.